Hi, my name is Fabio Madeira, and together with Matt Pierce, we are presenting today's technology track about EMBL-EBI Search and Sequence Analysis Tools APIs and their role during the current COVID-19 pandemic. So, Matt and I are project leads at the EMBL-EBI, and we work within web production. Web production is added by Rodrigo Lopez, and in web production we provide web infrastructure and core web services. So, two main core web services that we provide, two main projects are EBI Search, which is a full-text search engine for EMBL-EBI data. This part of the talk will be presented by Matt Pierce, and I will be presenting the other part about EMBL-EBI Tools framework. This is a sequence analysis tools framework that provides access to bioinformatics applications. So this is the outline of the part of the talk that I'm presenting, and I will give you an overview of what Job Dispatcher is, which is the tools framework, and I'll give you a summary of available tools and databases, an overview of the API, and give you an update on the relevance of JD during the COVID-19 pandemic, and just leave you with some useful links. So, as I mentioned, Job Dispatcher, the tools framework, provides access to core bioinformatics applications and their related up-to-date biological databases. So, just give you a list to give you an idea of the types of applications we provide. Provide applications in a number of different categories, for example, multi-sequence alignment, sequence similarity search, protein functional analysis, and other utilities. So, in terms of how one of the user interface web interface looks like, this is for cluster Omega how the web page looks like, and you can see there's a form. You need to paste your sequences of interest or upload a file, and you can select a number of different options and parameters for cluster Omega, so you can execute the sequence analysis and get your results output back. In terms of biological databases, some of the tools require those, for example, sequence similarity search tools as well as protein functional analysis tools. So these are, for example, NC-BioBlast, FASTA, or InterProScan, and we provide protein sequences, nucleotide sequences, and other specialized databases for some of the tools. As you can see, ranging from a number of different EMBL-EBI data resources such as UniProt-KB, ENA, Ensemble Genomes, and others. Again, this is how the user interface, the web user interface, looks like for another tool, in this case NC-BioBlast, and the difference really is that in this case you have an option to select biological databases. So, as I mentioned, we provide access to core bioinformatics applications, and we see this as a software as a service because in fact we provide programmatic access to the tools, so you basically can just use our services and instead of basically maintaining the tools or the databases on your own machine. So you can just use web services that we provide to access these bioinformatics applications yourself. So here now I give you an overview of the application programming interface, the API, so you can basically use the services yourself programmatically. So in this case, if you add to this web page, you can select among the different tools available, and you will see a page like this where you can have a look on different endpoints in the API. And I have to say that the API is actually the same for all the applications, with obviously some outputs of the endpoints are actually quite different, depending on the tool that you are using. So the first available endpoint here that you can use is /parameters. So in this case you get a list of available parameters for a particular tool, in this case cluster Omega. And from the list of available parameters you can then actually learn more about each individual parameter. So if you use the second endpoint here and use the name of the parameter, you can basically then get to know more about the parameters, so a description, the types of values it accepts, and also sort of the ranges and some other information. So the other endpoint here, submit job, is quite an important endpoint. So this is the post endpoint where you submit your job. So in this case you will require some input and some selection of options so that you can submit the sequence analysis that you want to run. When you successfully run a job, you will get a job identifier back, and with that you can actually get to know the status of the job, whether it's running, whether it's been completed or whether it's actually failed for some reason. And with that job identifier you can also get to know which types of outputs you have available for that particular job. And from that list of available result types you can then also obtain the actual output using this endpoint as shown here. In this case you need to replace the job identifier that you have and the type of output by its name. Just to give you an overview as well here, that if you expand in any of these endpoints you can actually try them, and for example in this case we are looking over the submit, the post request. In this case you would require to pass some sequence information in order to be able to try out the endpoint. Another example here for the first endpoint / parameters, where you can actually see once you have tried the endpoint, you can see an example of a curl command that you could just copy paste and use, execute in your own command line or terminal, and this is the type of output you would see. So because the API is common, is the same for across different applications, and it's easy to integrate in sequence analysis pipelines and workflows, and for that we actually help you by providing example clients to the API in Python, Perl, Java, and we also provide Docker images with those clients pre-installed, so you can use them out of the box if you have Docker runner in your machine. So in addition to that we provide CWL, which is Common Workflow Language descriptions for the services, for the applications, and we have also some example workflows that you can try to use to, for example, adapt for your own sequence analysis projects. These are just examples of integrations from popular resources at the EMBL-EBI. This goes to show the trust in our APIs. So the likes of UniProt, Protein Data Bank in Europe, Interpro, and Ensemble Genomes use our BLAST and all the multiple sequence alignment tools via our web services. And now to give you an update on the usage of the tools during the current COVID-19 pandemic. As you can see, over the last year, this is on a month by month basis, you can see that April and May there were quite a spike in the number of jobs that were executed, over 60 million jobs in April alone, and we run around 600 million jobs throughout in the entire year. And we can see that a lot of analyses were performed around COVID related sequences, doing phylogenetic analysis, multiple sequence alignments, as well as sequence similarity search on those types of sequences and on those types of analysis. And this was actually followed by a spike in the number of citations for the paper that describes the work that we are presenting here today. Just to wrap up my part, these are just some useful links that you can basically pause the presentation and just add over to in order to be able to get more about the services, documentation, and the clients and CWL workflow. So I will end up now to Matt. We'll continue with EBI Search part of the talk.
Hi, my name is Matt Pierce, and I'm the technical project lead of the EBI Search team, part of web production alongside Fabio's team. And today I'm going to be talking a little bit about EBI Search. We'll go through what EBI Search is, we'll take a quick look at the EBI Search API, and then we'll look at how the COVID-19 data portal uses EBI Search. So EBI Search is a full-text Lucene-based search engine, allowing search across multiple datasets at EMBL-EBI. The search results are always up to date. We index them every night. And in addition to the web application, we provide a RESTful interface allowing the EBI Search functionality to be integrated into other projects. The EBI Search web application actually uses the EBI Search API on the back end. In terms of statistics, we have nearly 4 billion biological entries available across 110 datasets, and we have had 500 million requests from over 970,000 unique hosts over the last 12 months or so. The API itself provides a number of categories of interface. You can search across single or multiple resources across the dataset. You can do cross-reference searches, finding links between resources or entries within those datasets. We also have autocomplete, top terms for getting popular terms from specific fields, sequence analysis tool result search, which allows you to search using sequence analysis results from external tools, more like this. And for a number of resources, we have raw data retrieval available. This is the EBI Search web application. You can see we've done a search here for TP53. We've drilled down through our sources to the UniProt KB dataset. And on the right-hand side here, you can see we've got a list of entries with fields, cross-references, formats that you can use to view the results. And on the left-hand side here, we have filters and facets allowing you to drill down and refine your search to make finding your results a bit easier. The same search retrieved from the API, presented here in XML format, although we do provide results in JSON or tab-separated format as well if you need them. You can see here at the top, we have the number of hits for our search. We have a list of entries. This is the basic metadata, but you can also ask for additional fields to be returned as you need them. And in the bottom half of the window here, we have filters and facets with the status facet expanded here. And so you can see the facet value and the number of entries. One of the most powerful features in EBI Search is cross-reference searching, which allows you to find the links between resources or entries. You can find the resources referenced by a resource on the left-hand side here. We've done a request for cross-references between UniProt and other resources, and you can see we reference Array Express repository, Atlas experiments, Atlas genes, and so on. You can also drill down and find resources that are referenced by a specific entry. We've done a search here for P53 human and looked up the cross-references for that. And you can see we've got a single results in Atlas genes and Atlas genes differential, 25 in biomodels, and so on down the list. Finally, you can do cross-reference search at the entry level. So in this particular example, we've looked up P53 human and we've asked for the cross-reference IDs from Europe PMC. And you can see here in the results list, we've got the IDs for 193 references in Europe PMC. The cross-reference searching is bidirectional. So if we were to cross-reference between UniProt and Europe PMC, as we did in the previous slide, we could then actually do a cross-reference lookup between the entry in Europe PMC and the corresponding entries back in UniProt to get the results there. The COVID-19 data portal is one of many sites across EMBL-EBI that use the EBI Search API. The other sites include ENA, RNA Central, Interpro, Metabolites, and so on. COVID-19 data portal uses the Search API for up-to-date content, cross-reference searching, and versatile and hierarchical search. There are a mixture of datasets in the portal. Some of them were specifically curated for the portal, while others are limited by querying to COVID-19 related content. In terms of innovations between EBI Search and the COVID-19 portal, there have been innovations on both sides, particularly on the UI. There have been UI changes in the portal that we've re-implemented in the EBI Search, and as we've developed those, they've been pushed back So you can see here we've got an association score facet as it was presented a couple of months ago, with checkboxes indicating the ranges available. And this is how it looks today. We're using a slider, and we've got a histogram display across the top here indicating how big each range is as you drill down. We've also implemented some data processing changes in EBI Search to accommodate the data for the portal. The portal has its own search API. It's an abstraction of the EBI Search API, limiting the results to the portal content. It's also simplified, so you can't do cross-reference search and so on. You can just do simple query searches. But you can, of course, query the same data sets directly through the EBI Search API if you need to. There are a close relationship between Job Dispatcher and EBI Search. So we enable sequence similarity searches across several data sets. You can see here we've got a UniProc KB result, and if we click through to the BLAST sequence search, we can do a similarity search here, and the selected result is already filled in the form. So here are some useful links. I'm not going to go through them, but particularly our RESTful API documentation is useful if you're looking up the EBI Search API. And I'd like to thank the rest of the web production team and my colleagues at EMBL-EBI. If you do use our services, please cite our paper. And any questions? Thank you.